Okay, thanks to everyone for coming. I understand it's toward the end of summer course. Today I'm going to share some of our research experience in terms of using a sparse representation-based biomarker identification for schizophrenia. But I'm just using schizophrenia as an example because we happen to have some data, both real data sets as well as simulated data set. But because of the reasons you understand, sometimes I cannot share the raw data in this talk because it belongs to collaborators and we have agreement. But we're doing our best to show some of the real data look like. So in the beginning, I just want to talk about our concept of data. So we all know these words, big data now. So in our group, which is like a core lab of statistical genomics, as well as data analysis, we actually handle all kinds of data, even though essentially we are more familiar with genomic data, such as single nucleotide polymorphism, RNA expression, or protein expression. But after we joining NIMH for 5.5 years, and we start to learn different things, we learn a lot from Dr. Koch's lab, such as neuroimaging. So many thanks to his lab, and including Rick, and help us a lot with quality control. Then we also learned a lot from clinical staff members at NIMH, so we're more familiar with how complex the diagnosis of schizophrenia as a phenotype, many other things, or even EEG, cognitive measures. So it's a very exciting stage of my research life. So today, I think I would just focus on two things. One is both task-based fMRI as well as resting stage ones, and some GWAS data that I'm most familiar with. I will show some a little bit ambitious integration algorithm we're proposing, what it can do, what it cannot do, as well as some potential limitations. So regardless to say, schizophrenia is one of the most chronically disabling psychiatric illness. Global-wise, the median lifetime morbid risk is about 7.2 over 1,000 persons, and both genomic and the brain imaging data can be used to explore the pathogenesis of schizophrenia. So as I mentioned earlier, this is very much a data integration algorithm. So two types of data. One is the GWAS and just SNPs, and then the other are two types of fMRI. One is resting stage, the other one is tasking with stimuli. So what is our goal? Obviously, we like to take advantage of potentially complementary information. In this regard, we're really just getting our feet wet, and we want to give a go, see what it turns out. And then the ambition is to seek higher power to identify potential biomarkers, if I may use that term, biomarkers, that could be missed when you only use one type of data. And that I refer to either GWAS SNPs or just fMRI data. See if we do two together, can we do better? If better, how much better? Okay. So genomic data, for the folks may not be very familiar with that. Basically, we have wealth amount of genome-wide, we call single nucleotide polymorphisms, and then you will see GWAS a lot across scientific journals because a lot of study have been done. And these GWAS studies typically focus on associations between SNP and human diseases, and they do not even have to be diseases. Could it be some kind of condition or BMI, right? Then also, the GWAS has been viewed as a powerful method in terms of identifying susceptible genes for common disease, but schizophrenia, bipolar, major depression, etc., that I would not say it's very powerful yet. The one part of the problem is the disease etiology is so complex. Even if we look at genetic alone, it's not like old Mendelian days. A single gene could be directly linked to a disease phenotype. Schizophrenia is just not like that. There must be many underlying genes and contribute not at an equal amount. On top of that, there's life events, environmental factors, many other things are related. So this, I want to emphasize, it's a data integration study, but by no means it's like looking for like association or something like causal effect, far from it. Okay, so I also want to mention the GWAS limitation a little bit, because if you look at one SNP at a time, and given that they contribute very little to disease, particularly to schizophrenia, so single point association analysis does not have a whole lot of power, which is also motivates this kind of analysis with integration, not like focus on one SNP at a time. Also, a lot of coded signals, they're not like causal, and at most it may be linked. And then at times, we always feel we need a much larger sample size to do a particular study, but we all, we don't have adequate amount of funding to do like the larger study if possible. But in a way, we're getting there. I mean, there are exciting news, maybe not particularly with schizophrenia, but I'm aware of one study which was led by Broad Institute, and they found, I believe, 108 genetic loci linked with schizophrenia. I can't use the word causal, but at the statistical level, they reached the gold standard, which is 5 times 10 to the minus 8. So I respect that in the statistical sense, but I'm making no comment whether these are functional variants or not, because that you need to move to the cell level and to prove something is causal. All right, so enough background information. And now just a few words about neuroimaging. I think the audience probably know more. As I insinuated earlier, 2 types of data will be presented here. One's task-based, the other one is the resting state, fMRI. Also, the goal, these are tools, and we're familiar with. They can be used to identify both structure as well as functional abnormalities in different brain regions. Sure.
You just talked about the difference between linked and causing in terms of genetics. Can you a little bit clarify that? How could it be linked but not caused by genetics?
So linked, I actually use the word associated. So it's like a linear regression model. So you establish association, but mainly you use p-value to judge whether something is significant or not. But it could be linked. I could give you an example. Let's say in an area, there is a true causal guy, okay? But in the neighborhood, you could have up to 10 or 20 SNPs. They're not causal at all, but they happen to be in the neighborhood of a rich person, and they got picked up. So in that case, it's just coded associated, but has nothing to do with causal. Does that make sense? Okay. All right. So here's a little math, and I do apologize put so dense, but otherwise I cannot to explain the regression model that we're proposing. I will use that less numbers and more English, hopefully, to clarify. So to some extent, it's just a multi-variable varied regression model. But the way we spelled out, it's actually easy. So it's alpha 1 multiplied by A1, and then the second term is alpha 2 by A2. As I started to talk about in the very beginning, we have two kind of phenotypes here. And the one is the neuroimaging data, and the other one is genetic data, right? So that's why we have A1, A2. On the other hand, we want to use coefficient because we want to do estimate, and we want to figure out to what extent these data are important. So that's why there's alpha 1 and alpha 2, and they adds up to 1. So sometimes we also call that as like weighting factors. So imagine you have two factors competing with each other. So that's what we try to do. It's like put them together independently or jointly together, see how many features they would each pick, or jointly how many they pick, and then using that to see it's a explore, to see how well the algorithm we're proposing work or not work. So, and then that involve the dimension reduction procedure. We starting with matrix. A1 is like n by P1 matrix. A2 is n by P2. And these measurements of two types of data. And I just say that one more time. One is the SNP array. There's a lot of SNPs. And the other one is the functional RNA data. As you know, it's also a humongous amount of data. So as I said earlier, alpha 1 plus alpha 2 need to equal to 1. They both need to be stay positive, and they are the weighting factors. And here's like an error term, and that is just a measurement caused by noises. And we're familiar with from regression analysis, right? So no need to go further to that. And then, sorry. So then I want to say a little more about how do we get weighting factors, as well as how do we do classifications. So that equation is showed again. So to determine this alpha 1 and alpha 2, one possibility to use cross-validation, and that has been very often used in dealing with large data. So in this particular case, we use cross-validation so that the weighting factors that generate the best classification rate. So this is our goal. Then what we did was the cross-validation, particularly we do the tenfold, and then 90% subjects from both cases controls were randomly selected for either variable or biomarker selection, and the rest were used for testing. I would assume the folks here understand the concept of cross-validation. In the machine learning world, it is a key. So this algorithm we're proposing, it's a type of machine learning in my view, even though I did not put in the title. So for each method, we carry out about 100 runs. We could do more, but due to the speed of a machine, but we will do later. We carried out about 100 runs and then average of the classification ratios, and that was used as the final indication of accuracy. So this is just a cartoon. Just want to acknowledge it's not my original drawing. It's coming from a paper called Assessing and Tuning Brain Decode. So that's viewed as a very good reference for machine learning, for cross-validation. So it talked about leave one sample out. I think we're all very familiar with that. And then you can do your own partition, like 20% leave out, and then do 3 splits. Another 20% leave out, 10 splits, or 50. So we all have our own style, but cross-validation is what we used. And then we talked about the weighting factors. And then I also want to point out for different pair of weighting factors, different variable groups will be selected, and that will lead to different classification ratios. And this is what we're going after. We want to know how well we can classify things. So this is an illustration just to simplify what we did. So y equals to a multiplied by x. It was the original equation. So n refers to the sample size. Typically, as you know, for individual with schizophrenia case and without, typically we don't have like a large collection. Let's say normally collaborators approach me, typically have a few hundreds cases versus a few hundreds controls. But then on the other hand, for the neuroimaging, there's a lot. So we have an interesting question here. We're facing a situation when the measurements itself, which I call the P, is a lot larger than N. N is the sample size I just mentioned. So this was why we use this called sparse representation algorithm to get through that. So recall the process I showed. So you first initialize it by setting delta equals to 0. And then for the step L, you can randomly choose what I call K columns from the X matrix. X matrix is a N by P dimension. And then after you choose K, we construct a submatrix. And now it's a N by K submatrix. And then we label it with the index, which is I in the script L. So that's what we did. So the most important part is to following the LP minimization procedure and to find an optimal sparse solution. Okay, so that's very much a minimized procedure, but it's important to say this is subject to this condition hold on the right side. Otherwise, it's not valid. Then it's becoming like minimization procedure we're all familiar. So you just you update to this delta L over and over. And then where the delta L, index L, and the delta L minus 1, index L, denote the delta L entries in delta L and delta L1, respectively. That's the details of the algorithm. But if this equation come out to be greater than alpha, by the way, alpha is something we decide. We can choose a constant, and then you just update L by L plus 1. And then you go to the step 1 that I showed earlier. I mean, if people are interested, I can give them the slide. Otherwise, you set delta equals to delta L divided by L. It's like a new circle restarts. So how does that work and how well it works? So let's say this is the original data, and it could be the integrated data, let's say, has both SNPs or the neuroimaging data. And then through random selection, we got this rectangle. This is select data, or I can call that XL. So this is like a very long, narrow vector. And I have to tell you, many of these terms are actually 0, right? So this is what we got. And this is, but after that, so this is our selection, and then it leads to prediction, like we're using classification ratio to indicate it's good or bad way. I also want to mention, because it's a new method, we look at it into convergence rate as well as multi-scale property. They both looked very nice, and this is how it converges with different K. And then this is a multiple scale property of the algorithm we developed. The further details is in the Neuroimage paper. You're welcome to take a look if you want to further the discussion. But I want to present some data, because without practice on real data, algorithm means nothing. So application one, this is, we have some real fMRI data that come from Shanghai University. So what happened was there's a researcher fellow from that university stayed 6 months in our lab. So we learned together, and he shared some of his data. So that's where they come from. So the sample size is the same as the GWAS. It's like 92 cases and 116 controls. And these are the SNP number, and 153,594 are the voxels coming from the real data. So the GWAS was simulated and versus task-based imaging data. It sounded a bit odd, but that's what we have to live with because we don't have the GWAS data. They're working on that. So for today's talk, I have to present what I have. So the classification rate actually was not that bad. If you use SNP alone using the algorithm I just illustrated, it's about 83.1. But if you look at a voxel alone, it's only 63.1. Yet, if you integration the two together using the exact searching algorithm I showed earlier, it's a little better. It's like 89.7. But again, I want to emphasize, it's a bit disappointing that the SNP part are simulated. It would be great when we get it. Maybe within 6 months with the same collaborator, we will get the SNP data. So hopefully I will update you our new results. Second application, I want to do something a little different. Here, both data sets are simulated because I want to see in the best case scenario what the results look like. So the first application was half real data, half simulated. And here's both simulated. This time, we choose 100 cases and 100 controls, and then we simulated 10,000 SNPs. And secondly, we simulated like resting stage MI data. So in terms of features, that should be easy. It's just 116 multiply 117, and then divide by 2. So we got 6,786 features. And then the features from connectivity between and within 116 AAL brain regions, you can you can easily calculate a connectivity. It's just the sum of correlated VI and the VJ divided by total sample size. So in terms of correlation, we used standard Pearson correlation coefficient. And N here is the number of voxel pairs. So finally, we got 116 multiplied by 117 divided by 2. And then our goal is to extract this connectivity. This is just a picture. It's, I think, A is just showing the 116 AAL brain region. B is using resting stage fMRI data alone. And the C is a mix of the integrated, so that including SNP and resting fMRI stage. It seems that the integrated did better, so the C, in terms of the amount of features got extracted, is more than B. The reason I say that is because I put like little color here, yellow and the red. And the yellow ones has high value, and you see a lot more yellow in this picture of C, but a lot less in B. So just for simple illustration. Another way to look at it, maybe more clear instead of the last figure. This is even more intuitive. So when we use SNPs feature alone, we selected 301 SNPs. at that time, we got a classification rate of 91%. And then if we use resting stage fMRI alone, we got 154 features. So the classification ratio is a little better than using SNP alone. I thought it's more or less expected. Then if we use both data together, we got 195 fMRI selected, and then the classification rate jumped up to 98.9. But again, this is simulation data. I'm not saying our algorithm could do as well in the real data, because real data has a lot more noise, and that was one challenge. But it shows some promise that we can keep going in this direction with this algorithm, and then try to obtain more data. It's becoming possible from publicly available resources. So we're not going to stop here. So basically, this is the results on 3. It's very similar, but just a different way to show. Here I use graph, but I want to point out, it's sort of interesting. This is just a random noise, and then even that alone also seems to have a classification like close to 70. That was a little bit a surprise to me. But if I think about it, I think in a way it makes sense. It's similar in the GWAS study, among these positive findings that could also be just false positives. And then I use red to indicate fMRI, green to indicate SNP, purple to indicate like a mix of 2 types of data. It looks like the purple was on the top, red comes second, and green is third. But it's also interesting if you look at the x-axis, where it shows the number of variables, and that's going and descending ranked by the SRVS algorithm. There's one part, too sensitive mouse. There's one part, the green light and the red seems like matte. And that might be because the number of variables are relatively small in this point. I think it occurs in real data as well. This one, again, is similar to what I just said. It's a different way to look at it. So the first bar is the imaging using the SNP feature alone. Second is the integrated data, and where 41% SNP data-based feature came out, and 59 for neuroimaging came out. And the third one is the SNP alone. So if you look at it in terms of classification rate, SNP feature alone, the classification rate is 91.7%. And then fMRI feature alone, it's 154 features were recovered with a better classification rate. Again, use integrated data. 195 features were selected, and classification rate is 98.9. So this just gives us a little bit idea about how the algorithm worked. But as I said, particularly second data set, it's all simulation based. So we probably should anticipate more challenges with the real data, even the very clean real data. So, but it seems to me integrating both type of data are promising, and we can even think about further data beyond just neuroimaging and the SNPs, because the thinking behind it is there's some kind of correlation between our genome and the phenome. The phenome is like phenotype, joint. So neuroimaging just one type of it. It could be many other things. Let's say if there's, BMI, age, gender, or other way, like other type of clinical information. If it's the diagnosis is very good, it's worth a try to including multimodality data. So seems like, and then also the other thing I did not do, which I should have done, is I could have include both resting stage as well as task based neuroimaging data together and with SNPs. So that's something can be done the next step. So seems like the algorithm did it reasonably well, but particularly when the number of variables is large and the sample size is very limited. But we will keep going, and then we're seeking for real data and move forward. So now I'm going to take a break and take some questions. Yeah.
I had a question about this slide where you had the classification accuracy.
This one?
The one before.
Yeah.
So the number of variables apparently is affecting how the classification ratio is changing.
Yes.
I just was wondering if the number of subjects have any role in where these plots are kind of breaking, or it has nothing to do with that. Because you could see, I don't know, maybe I think you mentioned you had about 100 subjects.
Yeah, I think a larger sample size would lead more stable results. That's for sure. But for neuroimaging, I don't know about the other friends here. In my group, typically we get up to like 300 cases and 300 controls because I grew up in China. So naturally, I sometimes just give lectures, like a teaching summer school. So that's how I meet the collaborators. But none of them have something like 1,000 brains. I mean, I wish they do. And also, between hospitals, they don't like to collaborate. I made my best efforts. It was not possible. There might be IIB issues involved. I mean, I shouldn't say that, but that's the truth. You know, I don't really like small sample size, but I cannot just sit there not to. I mean, we're still learning from sample size, right? But possibly there's a lot of noises involved.
But in the sense of the number of variables that you like to include in the classification, do you have to consider that with respect to the number of subjects, just from a
Yeah, I
statistical point
think
of view?
in this
Because that
case,
might be an overfitting
could be.
problem. Okay.
Yeah, I agree. Thanks for pointing out that. Even though it's like a nonlinear, we're not seeing it, but in the background, yeah.
Okay,
Any other questions? Or anything like you think probably better than what we're proposing we should try? All right. If not, I guess I think I'm done with this talk, but email me if you feel more comfortable to discuss things. Thank you so much for coming.